Good morning, class. Welcome back. Today our discussion is over Chapter 4. And by now, you should have taken the time to view all of the videos on this lecture page so that we can now have a discussion on cell structure and function. This week in the lab, you'll be finishing up your wheatgrass project that you've been conducting at home and typing up your formal report according to the Lab 1 Process of Science handout found in this laboratory module here at the bottom in Canvas. Here our chapter objectives include recognizing cellular structures, and being able to list their function, sometimes there's more than one, comparing different cell types, including prokaryotic cell types, which include bacteria, versus eukaryotic cell types, which include plants, animals, fungus, and protists. So let's get started. So in order to visualize very small structures and learn about them, biologists often use the help of a microscope. Your ebook describes this hurdle very well in the section titled Seeing the Unseen. The light microscope is a common tool used in any biolab. It uses a bright field of light that shines up from the bottom through your sample towards your eyes so that we can visualize small life forms and structures that measure in the range of micrometers. That's 10 to the minus 6 meters. Write out on your paper what 1 with 6 zeros looks like. You may recognize this as 1 million. When we use a negative sign, it means 1 millionth, designated by the Greek letter mu. On this scale, you can see tiny viral particles at the far left and proteins are on the order of nanometers, abbreviated NM, which is 10 to the minus 9th, and an electron microscope is required in order to visualize these. And on the far left, we can see how tiny molecules and atoms are on an even smaller scale. We do not have equipment powerful enough to be able to see these, and these are measured in angstroms, an A with a little tiny zero hat on top. An important part of biological history is the development of the cell theory and how it ties into the the invention of the first microscope. Antony van Leeuwenhoek in the 1600s was the first to observe single-cell movements. He looked at animal and plant tissues, at mineral crystals, and at fossils. He knew the difference between live and inanimate objects. He was the first to discover blood cells. Now fast forward to the 1830s when Schleiden and Schwann, this was a team effort between a botanist and a zoologist, they found a unified theory, first proposed, called the tenets of cell theory, which state all living organisms are composed of one or more cells. The cell is the basic unit of structure and organization in organisms. All cells arise from pre-existing cells. These tenets of cell theory, you will likely be asked about one of these basic rules that defines life as we know it on an exam, so it's a good idea to commit these to memory. Let's move on and start looking at microscopic cell structures. Cells can be categorized into two major groups based on structure. A prokaryotic cell type is usually associated with a single-cell life form, like a bacterium. These cell types lack any nucleus or internal machinery that we would call organelles. These are on the smaller size and usually take more magnification in order to visualize them. Fossil evidence indicates that these life forms first appeared 3.5 billion years ago. Just for a reference point, the Earth is estimated to be just over 4.5 billion years old. Eukaryotic cell types all have a visible nucleus and membrane-bound organelles. These are much larger in size and can be found in single-celled organisms like protists or in multicellular organisms like plants, animals, and fungus. The eukaryotic cell has been shown to have evolved about 2.1 billion years ago. So that's about 1.5 billion years after the prokaryotic cell type. Here's a look at what you'd see under the microscope when you examine these cell types. Escherichia coli is a commonly found example of the prokaryotic cell type. In the lab, we have to stain these tiny life forms in order to understand them better. Since they lack many of the hallmark cellular features, we can use a more powerful electron microscope to magnify them and be able to see um, other cellular characteristics that we maybe wouldn't be able to see with our light microscope, here stained in pink on the left. When we use a powerful electron microscope, we lose the color but gain more structural definition, which is why the picture in the middle looks kind of gray. And let's not leave out the unique archaea over here in the lower right. This is a scanning electron micrograph, which can give you a more 3D image. These very tiny life forms that are only found on our planet's extreme environmental conditions. For example, volcanic vents deep in the ocean, or places like the Dead Sea with very high salt concentrations. We find life carving out an existence in high heat, salinity, and pH extremes, but only if they're an archaea. Cell types like this make this possible. If we compare this now to organisms with the eukaryotic cell type, we have a lot of diversity in this category. This includes plants and animals, as well as fungus and single-celled protists. These protists might be confused as plant cells. Over here in the lower right-hand corner, they're green, but protists are very active little pond dwellers that are able to switch to photosynthesis when they're not consuming food particles. More on this later. Okay, so now let's compare the details of prokaryotes to eukaryotes so that you can ace the first exam. It's a nice thing to know that all cells share 4 common components, and we can usually see them with the help of a microscope. Here are some live cheek cells photographed with a phase contrast filter, so these are not stained or manipulated in any way. They're pretty much in their natural state. I've labeled the 4 parts or regions of the cell that you should expect to find whenever you're in the lab working on our various experiments planned for this semester, or on an exam when you're presented with a photo of a cell. In this example, I've chosen to use the eukaryotic cells harvested with a toothpick from a human mouth. Remember, eukaryotic cells are much larger and easier to identify. So let's go through the numbers, working from the outside to the inner structures that are visible to us. Number 1, the plasma membrane. It acts as an outer barrier that separates the cell's interior fluid compartment, often labeled as ICF, from its surrounding fluid environment, often labeled ECF. It's important to understand that this barrier is semi-permeable to different molecules or solutes. That means the cell is selective as to what comes in and out of the internal or ICF compartment. As a reminder of the structure or configuration of the plasma membrane, a diagram from the previous chapter is provided here just below our micrograph of the cells. The plasma membrane is unique and very important to the survival of cells. Here you can visualize the phospholipid bilayer, where the fatty acid tails are pointed away from the watery environments on either side of the cell membrane, and the phosphate circles, which are water-loving or hydrophilic, they're pointed towards the ICF and ECF compartments. Phospholipids display dual solubility and are defined as amphipathic. You may need to review your chemistry of life notes and Chapter 3 macromolecule notes before going any further. Number 2, the cytoplasm. This is found in the ICF compartment only and consists of a jelly-like cytosol that suspends many of the cellular components and organelles. This area is often just labeled as ICF, which stands for intracellular fluid compartment. Number 3, the nucleus. A centrally located dark staining organelle that contains DNA, which is the cell's genetic material. The nucleus is the most prominent of all the organelles that we're going to learn about. This highly visible organelle can help us when we're trying to focus the microscope on a cell in the lab. And number 4, if we go back and look closely at the ICF compartment, we can see that there's some stuff suspended inside the cell, and this is where another important type of organelle can be found called the ribosome. All cell types, whether prokaryotic or eukaryotic, need ribosomes to synthesize proteins. These organelles can look like little polka dots in the ICF compartment, and they lack a membrane, which is characteristic of all the other organelles we're going to study here. So let's start with the simple prokaryotic structures and some of their function. Remember, prokaryotes are different from eukaryotes in many ways and are 1.5 billion years older and more primitive than the more complex eukaryotic cell types we just looked at. Prokaryotic cells are much smaller and need a lot more magnification in order to visualize them, and we must use special stains that bind to the cell wall structures in order to even see these little guys. You will never see a nucleus in these cell types, but they do possess some specialized structures that help protect them since they are so tiny. Labeled for you in this diagram, you'll find an outer cell wall that protects the cell membrane. Sometimes there's even an outer sticky capsule. These spiky protrusions, called pili, they can help with attachment to other structures. There's also a whip-like tail here at the end that will propel these tiny organisms through their environment. In your full set of class notes, you'll find a vocabulary list that will help you in your study of these prokaryotic cell types. When we consider the eukaryotes, this large, diverse group includes plants, animals, fungi, and protists. Protists are the single-celled eukaryotes that live in water, and we'll want to do some further comparisons in order to understand this diverse group a little bit better. So when comparing plant cells to animal cells, we can appreciate certain similarities like the presence of a plasma membrane, the inner cytoplasmic compartment, or ICF, a nucleus in the center, and the polka dot-looking ribosomes inside each of these cell types. They share many of the same organelles, which we will cover in more detail next. But the plant cell, it possesses a cell wall, like we saw in the bacterial cell types, and the presence of a cell wall is going to help stabilize the plant during times of hydration and dehydration, depending on the presence or absence of abundant water. The large central vacuole over here on the right, found in plant cells, also helps stabilize the cell structure during times of drought. And the presence of green chloroplasts, these allow plants to carry out photosynthesis. These three main differences found at the cellular level make plants unique from their animal counterparts in the domain of eukaryotes. A closer look into the inner cytosolic chamber of an animal cell reveals the multitude of organelle machinery working inside these complex cell types. We will discuss the structures and function of each of these individual membrane-bound organelles next. Under the microscope, these plant cells all contain a large water-filled vacuole in the center of each cell. This oversized organelle tends to push the green chloroplast out towards the outer edge of the cell. Students are responsible for this list of organelles highlighted here in red. Your goal should be to be able to describe each of these with regard to structure and function, and it may be helpful for you to draw these on a piece of scratch paper and keep it in your notes when you go to review for the first exam. Each organelle structure has its own job to do for the cell. For example, ribosomes help manufacture proteins needed by the cell. Sugars and fats are also managed here inside the cell. As you study the inner structure of animal cells, you'll only have to make a few adjustments for the plant cell. We all know plants are typically green, and that is due to the presence of chloroplasts. We've also seen the large water-containing central vacuole, and all plant cells will have a cell wall, like we saw in the prokaryotic bacterial cell types. These three additions should be included in your study of the plant cell. Eventually, we will do our own observations in the lab using leaf tissue, where we can see for ourselves these important plant features. Now let's go back and build a eukaryotic cell from the beginning, starting with the plasma membrane, sometimes labeled as cell membrane. This structure is a common feature to all cell types, and in some cell types like bacterial and plant cells, the cell membrane is protected by a cell wall. We've already covered the fact that the cell membrane is made up of a phospholipid bilayer that helps regulate the passage of molecules into and out of the cell. The cell membrane is very fluid-like. Have you ever watched a lava lamp warm up? We will cap our Unit 1 studies with Chapter 5 and focus all of our attention on the movement of molecules across this structure. So this is mainly just an introduction to all of the cell parts. Zooming in even closer, The plasma membrane is not just a simple phospholipid bilayer, which is in the diagram here on the left. The bilayer is separated so that you can see the two halves. When we look closely, we can appreciate the presence of large functional proteins highlighted in purple. These are inserted into the membrane. Some large proteins are completely integrated into the membrane and are exposed on both the ECF and ICF compartments of the cell. Other smaller proteins are only associated with the inside ICF compartment, and these proteins are called peripheral proteins. On the outer surface, we see strings of sugars bound to proteins. These are used to identify blood types and used during cell-to-cell communication. By incorporating functional proteins into the cell membrane, the cell gains better control of the movement of molecules into and out of the ICF compartment. Inside the cytoplasm, the ICF compartment houses this fluidy, jelly-like matrix and all of the organelles on your list of study. This is a fluorescently stained micrograph of the cytoplasmic compartment, and it illustrates the matrix of proteins found inside the ICF compartment. So we call it a watery world because most organisms are mostly water, but we can see there's a lot of structural proteins involved as well. Now we can take a closer look at the nucleus. You should be able to easily identify this structure when magnified under a microscope. But there are some important structures we should consider, and these include the nuclear membrane, nuclear pores, and the nucleolus. Here in this diagram, we can look at just the nucleus, and we can see that the membrane is actually a double membrane structure that forms what's called an envelope, and it protects the delicate DNA chromosomes inside. The darker core in the middle is the nucleolus, where the ribosomes we mentioned earlier can be manufactured from what's called rRNA. That stands for ribosomal RNA, and we'll come back to this in Unit 3. Here's a look at the ribosome. This is a computer-generated image. It shows a unique quality to this organelle because it's the only one that lacks its own membrane. The ribosome is made up of two subunits, highlighted here in different shades of purple. These two halves will assemble once protein synthesis begins, also a topic for Unit 3. Ribosomes are found in both the free or bound form, which means they can be attached to another organelle's membrane, forming the rough endoplasmic reticulum, but they can also do the work of protein synthesis in the free-roaming unbound form. As a point of reference, this diagram is provided so that you can see the intimate connection between the nuclear envelope and the transition into the structure riddled with bound ribosomes that forms the rough endoplasmic reticulum, abbreviated ER. This organelle is committed to making nearly all the proteins required by the cell. These are made on demand and at the direction of the DNA housed in the nucleus. There is a smooth version of this maze-like organelle, which means it lacks the ribosomes responsible for protein synthesis. The smooth ER carries out very different functions, such as calcium storage used in muscle functions and the manufacture of certain hormones and lipids. As we move out away from the nucleus near the center of the cell, we find an interesting stack of membrane-bound organelles called the Golgi apparatus. You may see this organelle listed as Golgi bodies. You can think of this piece of machinery as the UPS of the cell, because what happens here is the delivery of newly made proteins, where they must get sorted, packaged, zip code attached, and delivered to the correct address within the cell. Sometimes proteins are exported or expelled from the cell, and other proteins must stay inside the cell to do their work. The Golgi apparatus takes care of all of this. When we get into transcription and translation of proteins in Unit 3, you'll learn a lot more about this organelle's function. The mitochondria. These are considered the powerhouses of the eukaryotic cell type. Here is where a universal form of energy is produced called ATP. This organelle is going to be a major focus as we move into Unit 2 in just a couple of more weeks. Housed within this double membrane organelle is another series of membranes that form what is called a matrix. These are highly folded and look very convoluted. This allows for maximal surface area where there's a series of protein enzymes that perform a controlled sequence of chemical reactions involved in cellular respiration. They use oxygen to harvest chemical energy from sugars, primarily glucose. Ultimately, this harvested energy is stored as chemical energy in the form of ATP, a more universal energy form, which can then be used to power many other cellular activities. Here are a couple of lesser talked about organelles, and I'll finish with these. These are the lysosomes, meaning lyse or to burst, and the peroxisomes. Maybe you've used peroxide before and have observed the foaming action. These are both important when we consider the inside of any organism needs to have some defense mechanisms in place to ward off any infections caused by foreign invaders like parasites and bacteria. These small but mighty little organelles contain some nasty chemical enzymes like hydrolase and peroxidase that will destroy foreign invaders. Well, we have covered a great deal of cellular structures and a little bit about their unique function in this lecture. There are a lot of vocab terms you will have to master, and your homework assignments will help with that. But you should finish up on your own with the terms highlighted in green on the very last slide in this note set, which covers storage and locomotion vocabulary terms. Many of these cellular structures and their function will be covered again in future discussions throughout our semester in biology. As we move forward, I'll operate with the understanding that you have mastered the material in this extensive chapter on cell biology. Okay, see you next time.